One of the new tools that's come out through genetics is something called the barcode. And just as when you walk into a grocery store and you see a barcode on the food that you may want to buy, we can actually create barcodes for different species. And this utilizes a particular gene of different species. And if we look at that gene, we find that that gene is very consistent within species but varies among different species. And so we can use this to try and tell apart things that are very difficult to identify. One of the most interesting stories I thought that came out of the census was actually a study two high school girls in New York City did, where they went out and sampled fish markets and restaurants in New York City and did barcoding on the fish that they collected from those excursions. And what they found is that a lot of fish was being sold under different names than what it was being billed at. So for example, we have things being sold as white tuna, which cost $8.50 a pound, which was in fact tilapia, which is worth $1.70 a pound. And my colleague, a guy named Dirk Steinke at the University of Guelph, has said to the census community, pretty much anywhere you go out to a restaurant, at least in North America, if you bring back some of those fish samples, some of them are going to be erroneous. There is going to be misidentified fish no matter where you are. Now, the reason this is very important is not just because of people cheating on what they're selling you in the way of fish, but some of these fish actually were endangered species. And it also allows us, again, to differentiate things that are very difficult to tell apart. And so we can monitor catches and make sure that things are being removed that we think are being removed, even if they look like similar sorts of species. Now, one of the things that we can do is to try and make the most of what we have, try and amalgamate the knowledge that we have about life in the oceans to try and improve our understanding, and better yet, to manage the oceans more effectively than we have in the past. And so this is a graph that shows you data that's been collected from the global oceans and put together into a single database called the Ocean Biogeographic Information System. The main points to take out of this graph, if you look at this area in yellow around Europe, that shows you the most intensively sampled areas of the world. The blue and the black show you the most poorly sampled areas around the world. So if you look at Eastern North America, not too bad. Europe, very good. Pretty much the rest of the world, very poor. In the inset, you see a cross-section of the ocean. And so at the top of this figure, you would see the surface of the ocean, and then at the bottom is the ocean bottom. The area in blue shows you that this is almost a completely unsampled volume of the ocean, which in fact is one of the most large habitats of the ocean, and we have almost no data from it. And so we have a lot of work to do. Now, what we can do with the data that we have is to try and figure out where the hot spots are. And the reason we might want to know about that is because if we're going to prioritize areas for conservation to try and minimize our impact, then one of the criteria we might use is to avoid areas that are very diverse. And so this is work that recently came out within the last year and shows that for taxes that live in the coastal area, it's really the tropics that are the most species rich. And this is very consistent with what we've seen in the terrestrial literature, very much a tropical species rich environment. But if we look at oceanic taxa, in fact, more temperate mid-latitudes, for example, the sort of latitudes we have here, are among the most species rich. Why do we care? What do all these species do out there? For most, we don't know. There are a variety of studies that are starting to address this question, but we tend to think of the oceans as a source perhaps of seafood, but in fact they do a whole lot of other things. We do not know the role that biodiversity plays in that process. We know living organisms are important, but less confident about biodiversity. We can also map the oceans much more effectively than we could in the past. I mentioned earlier that we have remotely operated vehicles like ROPOS. We have various acoustic instruments that bounce sound waves off the seafloor. Those sound waves bounce back up and are listened to, and based on the way that they bounce back, tells us a lot about the composition of the seafloor. And so now we can produce very detailed maps of what the seafloor looks like, and that is also a key aspect of how we might go about managing the oceans more effectively. If we know where the habitats are, we can start to make sure that we at least conserve the habitats, even if we can't be certain that we're conserving all the species that they contain. One of the other exciting technologies, and this is very important from an ocean management perspective, is the use of sound waves to count fish. And the idea here is that you use a device that produces sound waves, it bounces off the fish, it doesn't hurt them, it reflects back, and it tells you how many fishes are out there. And so what you see here in this image in the upper right-hand corner is an area of fish. It's a large school of herring that are about the size of Manhattan Island. And it's actually, yeah, these are herring. So it's about the size of Manhattan Island. And this was a set of data that was gathered in less than a minute using these sound waves. Now, traditionally, we would have had to go out there with nets. We would have had to collect the fish, identify them, and it would have been much more destructive and much more time-consuming. And so if we can get better estimates on how many fish are there, then we can also make better extrapolations of how many we can afford to extract before we start to cause the population to go into decline. Vancouver Aquarium, hopefully many of you got to see the display, is the very location for the Pacific Ocean Shelf Tracking Project, which is one of those 14 field projects that I mentioned at the beginning. And the idea here is that they put up arrays of listening stations which can hear little tags as they pass by. And so what they do is they surgically implant these little tags in salmon, and then they're able to see where the salmon move up and down the coastline. Now, I know in 2009 you had a disastrous year for sockeye salmon. In 2010 it was a spectacularly good year. Well, one of the things that we know is that the majority of these sockeye salmon that migrate out of the rivers that you see here on this image come up and move through the Johnson Strait towards the north. A relatively small number seem to come down and go through the Strait of Juan de Fuca. As it turns out, the survivorship of the ones that go out through the Strait of Juan de Fuca is much better than the ones that go to the north. We don't know why that's true, but nonetheless it is true. So if we can start to learn where the salmon are dying in the ocean, then that can make us better managers because we can start to try and do something about that. We know that some of our very early use of the oceans began with the Romans. And if you think about it, one of the things that would have constrained extraction of resources from the oceans is the lack of preservation techniques. We did not have refrigerators at that time, and salting had not been invented. So all you could do was catch fish that you could consume right away. Now, if you wanted to sell it on a market, you could perhaps buy yourself a couple of hours by going out early in the morning, but you still had to sell it that day or else it would rot. The Romans invented salting, and once salting was invented, that opened up the potential to transport fish, to store fish, and to catch way more fish than you could consume. And so thus began industrial fishing. We can look at a whole variety of sources to try and piece together what those oceans looked like before we as scientists were able to start sampling it. And in this middle image that you see here on your right-hand side, your left-hand side, is a painting from Franz Snyder, a Belgian painter. And I show it to you because it shows a lot of fishes that you do not find in the North Sea anymore. And we know that he actually painted very realistic scenes based on his other paintings. And so we can use these bits of data to try and understand what the oceans looked like before we were collecting science data. And so we know that these other species of fish used to occur in the North Sea, even though they no longer do. I like very much the image on your right-hand side, which shows you fishing derbies in Florida from 1950 through 2007. In the 1950s, you see some pretty impressive-looking fish. They're pretty big, a diversity of species. In the 1980s, they're getting a little smaller, a little bit less impressive. 2007, I would say that people in the 1950s would have laughed at these as trophy fish, and yet these were the winners from the 2007 fishing derby. And so we've changed not just the size of fish, but the composition of fish. And so this is data from Ransom Myers. This is actually the Gulf of Mexico, if I have the right data set here. And what we're seeing is that the removal of great sharks in the top panels is allowing small pesky sharks to increase in number, which is the second set of panels. What's interesting is the bottom panel, which you see in sharp decline in the last decade or so, is scallops. And this is what we call cascading effects of predator removal. So what's happened here is we've removed the top predators, in this case the great sharks. That has allowed lesser predators to increase in number, and those lesser predators actually go after the scallops, which then causes cascading decline. So this illustrates the point that some of our activities at the top of the food web have very serious ramifications further down the food web to things that we don't necessarily realize until we start to look at the data. Here's some more evidence of changing oceans. This is actually tuna and billfish around the world from the 1950s through the 1990s. In the 1950s, you see those hot spots of red, and so that's species density. How many different species of tuna and billfish were being caught in these average catches, these different efforts to longline. In the 1970s, you can see that the numbers are starting to decline, but also the effort is expanding. the fishing is starting to move into further waters north, further waters south. And by the 1990s, it's all very blue, so we don't see any of this intense diversity that we saw in the 1950s. And we also see that the fishermen are having to look further and further afield in order to find their fish. So I said I would say something about the future oceans. And the news is mostly not very good. I think when we look to 2020 and beyond, we can say with some confidence that the oceans will be more crowded. There will be more environmental changes because of that crowding. There's going to be more biological change because we are going to continue to remove species from the oceans because we have to feed the people that we have on this planet. There will be more ocean conflicts because this food will become more and more scarce and more important to different nations around the world. This is true for both living and for non-living resources. But the one good bit of news is that there's going to be better information. We are going to be able to see into the oceans better, and that's already true. So the pressures on the oceans are going to worsen, they're not going to lessen. And so my feeling is that there are three things that we really need to continue to do as scientists, and the general public, I would hope, will help us in trying to make this happen. First of all, establish a baseline of what's there. For some parts of the ocean, we have a baseline. We know what's there, but for many parts, we don't. The deep ocean is very poorly sampled, and for most areas, we have no baseline. The Arctic is very poorly sampled, and again, that's an area that's very likely to undergo tremendous change through climate warming. I think we also need to figure out which species really make a difference. We don't know what all those species are out there for. Do they do something? If we lose them, does the system become compromised? Or does it continue to hum along with or without those species? In the vast majority of cases, we simply do not know the answer to that question. And then finally, I think we need to provide managers with rational information to make decisions on how they're going to manage things in the ocean spatially. So if, for example, there's a decision to create a marine protected area, what is the purpose of that protected area, and can we give them the best information to make sure the location they choose will actually serve the function that we hope it will? We have a lot of ocean. We have the longest coastline in the world. We have relatively few people. We are not a very populous country, and relatively few scientists as well. We also have tight budgets. We're seeing cuts at DFO and elsewhere in the federal government. And one of the other quirks that's really relatively unique to Canada is that most of our population, with the city of Vancouver as a key exception, live on the US border. They do not live on the ocean. And so the problem we have as scientists and as people is to try and convince people in the 905-416 area code that they need to care about the health in the oceans. And so some of the people living in Toronto and Ottawa may never see the oceans, but they need to understand the importance that the oceans hold for every second breath that they take. So I think on that note, I will finish up, and I thank you for your attention, and I'd be happy to answer any questions that you have. Thank you.